Hello everyone and welcome back to microbiology. Today we will be covering chapter 11, part 2, Mechanisms of Microbial Genetics. In the previous lecture, we discussed that there are two functions of DNA, that of inheritance and that of being a blueprint. Last lecture, we focused on the structure of DNA and DNA replication. And in this lecture, we will be discussing how DNA stores information and how that information is converted into proteins through the process of transcription and translation. So let's look at the process of transcription and translation. That is, how the information stored in DNA is put into action. DNA is first transcribed. The process of transcription involves the synthesis of a complementary RNA strand from a DNA template. This RNA strand is then used for translation. Translation is the synthesis of protein from information encoded in the RNA copy. So we can see those two main steps as DNA being transcribed into RNA and RNA being translated into protein. Proteins are the workhorses of the cell. We previously covered how proteins work as enzymes, which control and regulate reaction rates, and they are largely responsible for encoding traits. So why does the cell use this process? Why doesn't the DNA just code for proteins directly? And the answer to this question lies in preserving the master copy. If I, as the instructor, only had one copy of the lab manual, I wouldn't loan out the lab manual to any particular one student to use. Instead, I would make copies of this lab manual to share with other students. So, the process of transcription begins with RNA polymerase, pictured here as this brownish glob. And what happens is the RNA polymerase binds to a promoter region, which promotes the transcription of a sequence. And then it unwinds the DNA strand, much like helicase does. It also reads the DNA base sequence, and recall that these enzymes are going to read in a 3' to 5' direction, and it's going to write or create a complementary strand of messenger RNA. So if we look at this GIF here, the composition of these nucleotide bases on the sort of the top strand that's being synthesized is what we call an mRNA strand. In eukaryotes, there is an extra step that occurs. This doesn't happen in prokaryotes. After transcription, that mRNA molecule needs to be further processed. This is a post-transcriptional modification that occurs, and this is where a splicing event happens. And there are two different types of sequences found in this mRNA molecule. One is called introns, and the other are called exons. All of the exons, noted as a red color in this image, are spliced back together. And the exons are the part of the mRNA strand that will exit the nucleus and further be translated into protons. The introns, demonstrated here as the yellow color, are cut out of the mRNA strand and removed entirely. And so the introns aren't really exiting the nucleus at any given time. So let's continue moving along. That is the process of transcription, and now we need to work out translation. So transcription is making just a script, almost like you could imagine a doctor writing a doctor's note in order to receive medicine. The medicine is going to be the protein at the end of this process. So In that way, we need to convert RNA into the information stored in RNA into a protein. Proteins are made up of a series of amino acids. So translation takes information in the nucleotides in order to write out a sequence of amino acids. These amino acids are strung together into a chain, and that chain is what becomes a protein. Here we have a variety of amino acids that could occur in living organisms, and these amino acids can be grouped based upon certain features they have. It's not a requirement in this class to memorize every single amino acid, but you should be aware of them, that we have nonpolar amino acids. And these nonpolar amino acids, as you can see, they have these hydrocarbon chains, and so they tend not to react with aqueous solutions very well. They do much better with binding to other nonpolar compounds. So this would be like lipid membranes, for example. We have the polar compounds, and you can see there's a lot of hydroxyl groups here, OH groups. Here we have a sulfur hydrogen group and oxygen groups. And these oxygens, these sulfurs, these molecules enjoy interacting. They're hydrophilic, and they will interact with water and with each other, with other polar molecules. We can also have electrically charged amino acids. So we can have negatively charged amino acids. Here we see these are acidic in nature, and these acidic groups have this what we call a carboxylic acid group, where you have a double bonded oxygen and a negatively charged oxygen here. We also can have basic groups. So these basic groups have a positive charge. You often see these as amines, or compounds, functional groups that have a nitrogen present in them. Again, there's no need to memorize these different functional groups or these different names, but be aware that there are three main types of amino acids. Lastly, be aware that for electrically charged groups, they can be either acidic or basic.
Checkpoint 1. Nucleic acids like DNA and RNA are made of the monomer nucleotides. What about proteins?
So, when the cell wants to make a protein from the RNA, what ingredients are necessary for this to occur? First, it needs the information coded on that RNA. This RNA is called a messenger RNA because it brings a message to the ribosome. It needs individual free amino acids to be delivered by a transfer RNA called a tRNA. It also needs the ribosome, which is the machinery in the cell that creates proteins. The ribosome is a large, complex structure of RNA and proteins that carries out protein synthesis. Transfer RNA, also called tRNA, are molecules that are bilingual. The amino acid is carried at one end of the tRNA, as seen here. Here's the tRNA, which is just made of RNA, and at one end it attaches to that carboxyl group of the amino acid, seen here. And this is called a ester bond. We've talked about phosphodiester bonds. Well, this is a ester bond yet again. Then it has a set of 3 RNA nucleotides at the bottom, shown as the anticodon. So this anticodon, as you can see, has a C, a G, and a G. And so a mRNA strand, if it's homologous to this anticodon, it will have the opposite. It will have a G, a C, and a C. As you can recall, A G always binds to a C, and an A always binds to a T or a U if it is a RNA strand. So cells have tRNAs for every amino acid / anticodon pairing. So as you can imagine, these little molecules will be used to read that RNA code, and if there is a match, it will provide the proper amino acid that can be transferred to the growing protein strand. So with the anticodon pairing up with the codon, we can see that the mRNA language is written in what are called codons, which are 3-letter codes that encode for a amino acid. We can use something called a codon usage chart in order to decipher this mRNA code and figure out which amino acids are coded for per codon. In this codon usage chart, you have the first letter of 3 here to the left, the second letter at the top, and the third letter to the right. So if we had the code AUG, we would look at A here at the first, then we would look at U for the second letter. So we know that it's going to be in this box here. The final letter is G, so if we go over to G, we see that AUG stands for methionine, and methionine is also the start codon. In every gene, there is a start codon, which tells the ribosome where to begin, and a stop codon. There are 3 different stop codons, which are UAA, UAG, and UGA, and that tells the ribosome where a particular protein ends. As you will recall, DNA in living organisms is double-stranded. The 2 strands are called complementary and antiparallel. That is because one strand, which would be written in a 5' to 3' orientation, has another strand attached to it that goes in the opposite direction, or the antiparallel direction, from 5' to 3'. They are called complementary because C's always bind with G's and T's always bind with A's through hydrogen binding. However, mRNA is single-stranded. The RNA polymerase uses one of the strands of the double-stranded DNA to make a copy into mRNA. This strand that is used to make mRNA is called the template strand. As the name implies, it's the template for making mRNA. So again, the template strand is used as a template for transcribing DNA to mRNA. The other strand of DNA is called the coding strand, and this is because it looks very similar to the mRNA strand. The only difference is that with RNA, you have a uracil base, and with DNA, you have a thymine base. This should intuitively make sense because, in a way, they are both antiparallel and complementary to the template strand, so they should have the same sequence. Geneticists have a writing convention for expressing the coding strand and the template strand. The coding strand is always written from left to right, from 5' to 3'. In that way, you can read this sequence as it would appear to the ribosome. Here we have the start codon, the ATG. Then we have the first codon after the start codon and many of the other codons. And then the last codon in this sentence is going to be the stop codon. And for that matter, if I gave you a strand and you needed to transcribe that into RNA, if you were looking at the coding strand, you would rewrite it the exact same way, only changing the T's to U's. As you can see, this ATG is a UG, and the next codon is a GT, which would become AGU. If I was to give you the template strand You could also derive the mRNA strand, but first you would have to take the template strand and create the coding strand. So TAC is complementary to ATG, and ATG turned to mRNA is AUG. And you can do this on and on down the chain until you reach a stop codon, at which that would be your final codon in this gene sequence. So let's now take an mRNA sequence and translate it into a protein. So again, the information contained in an mRNA transcript is organized into words which are known as codons that are 3 nucleotides long each. Each word corresponds to a different amino acid. So we can take any mRNA code, we can separate it into 3-letter codons, and then use a codon usage chart to see which amino acid each one of those codons codes for. In this case, AUG codes for methionine, which is the start codon. GUG codes for valine, CAC codes for histidine, CUG codes for leucine, and ACU codes for threonine.
Checkpoint 2. Using the codon chart, translate the following mRNA into protein. And here is the codon chart to be using for that.
Now let's reflect back on translation and see how this works in action. So that is the single-stranded mRNA code going through the ribosome. The ribosome is the brown globular-looking thing in the background. Each one of those little molecules that's shaped like a T are the transfer RNAs. Their anticodons are homologous to the codons of the mRNA. And that purple sphere on the top of them is that site where the amino acid is attached. Those amino acids are represented here as those purple spheres. And if the right anticodon finds the right codon, then the tRNA will stay in the ribosome pocket and transfer its amino acid to the growing chain. So there are some minor differences between how eukaryotes and prokaryotes do their transcription and translation. With eukaryotes, it's a little bit more complicated because they make first a pre-mRNA transcript, and this pre-mRNA, as we previously discussed, needs to have a post-transcriptional modification. That's where what are called introns are cut out and removed, and then the exons are attached back together. These exons are now ready to exit the nucleus, whereas the introns stay inside of the nucleus. So this mature mRNA, which is made up of just the exons, exits the nuclear membrane and is taken to the ribosome, where it is translated into proteins. In prokaryotes, there is not a nucleus and there is not the same sort of post-transcriptional modification that occurs. So there are quite a few of these genes that scientists have identified. For example, Staphylococcus aureus is a bacterium that has about 2,700 protein coding genes. E. coli, which we're all pretty familiar with now, has about 4,300 protein coding genes. And so, I would ask you, are all of these genes being transcribed and translated all the time? Go ahead and think about that for a moment. Intuitively, this doesn't quite make sense. You shouldn't need every protein on all the time. You only need to transcribe proteins when they're needed. So the answer is no. And what this comes down to is expression. Expression means is when a gene is turned into a protein, and so it is expressed or exhibited by the cell. There are two main types of expression. There are constitutive genes, and these genes are needed as a normal part of cellular operations. So they're just continuously being expressed, which means transcribed and translated. There's another group called facultative genes. Facultative genes are only expressed when they are needed. So they are expressed during response to certain environmental stimuli. Our best estimates are that about 60 to 80% of genes are constitutive and the rest are facultative. One way that the cell controls when to express a gene is through a process called pre-transcriptional control. Every gene is in a DNA structure called the operon. The operon is all the necessary components in order to transcribe a gene. It begins with a promoter. A promoter you can think of as a region that promotes transcription. What it specifically does is that it's a special sequence of DNA that the RNA polymerase can recognize and bind to. And if the RNA polymerase binds to the DNA, then it can initiate transcription. So no promoter, no RNA polymerase binding, and no initiation of transcription. Following the promoter is a region called the operator. The operator is a place that acts sort of like a traffic light for the RNA polymerase. The promoter and operator region is called the control region, and the region where genes are located is called the structural gene region. The two of them put together are called the operon. You can have several genes under one operon, but only one promoter and operator are usually present for an operon. And it looks something like this. You can imagine that this Cybertruck is the RNA polymerase. At the promoter site, it's bound, and at the operator, there is a traffic light. If the traffic light is red, then the Cybertruck will not go through and make a copy of that mRNA. However, if the light changes green, then the Cybertruck or the RNA polymerase will zip through that DNA sequence and make a mRNA copy from the template strand. Within an operon, you will find a group of genes that are related to the same task or same metabolic process. These all can be transcribed together and controlled by the same promoter. So again, we have the same control region that could control several structural genes. And the control region is made of the promoter and the operator, and all of this together creates the operon. There are two different types of operons. There are inducible operons and repressible operons. Remember that inducible operons are normally off. They are induced or they are turned on in order for expression to occur. Repressible operons are normally on and must be turned off. So again, inducible operons are normally off. Repressible operons are normally on. And let's go over an example of this. The most commonly studied or discussed example of this is the lac operon in E. coli. It encodes for beta-galactosidase, which metabolizes lactose. That is encoded by the gene Z. It has the Y gene in its operon, which encodes for a lac permease, and this transports lactose into the cell. Lastly, it has a transacetylase, which metabolizes other disaccharides, and less is known about the LAC-A gene. And all of these genes can be turned on together under the same promoter operator system. So in this image here, I've shown you a metabolic pathway map for galactose metabolism. Don't panic, you're not going to be required to memorize any of the enzymes here, but I just wanted to show you the complexity in these metabolic maps in order to break down a particular compound and make energy from it. So here's lactose right here, and you can imagine that there's going to be a lot of steps in this pathway in order to make energy or other usable products from this. So the cell doesn't always have lactose present or on hand. It's one of those sugars that might occur in the environment. It might not occur. And so the cell isn't going to make all of these different enzymes in order to do lactose metabolism, because that would require an unnecessary amount of enzymes in the cell. So if you were to ask yourself, if you were an E. coli cell, would you rather have lactose metabolism normally turned off or normally turned on? Hopefully you said you would want it normally turned off, because lactose isn't always on hand. It's not necessary to have it on all the time. And that's right. The lac operon is normally turned off, and it's turned off by something called a repressor protein. Repressor proteins can act on the operator to repress transcription. So normally this repressor protein is present. It's encoded by what we call LAC-I. So normally this protein is present because normally this operon is turned off. However, there are conditions, environmental stimuli, which inactivate this repressor protein. And that environmental condition is the presence of lactose. There is a metabolic derivative of lactose, and it's called allolactose. And this metabolite can bind to this repressor, this LAC-I repressor protein, and when it binds to it, it inactivates it. So here's the inactivation of the repressor protein, and we can imagine then if this bubble is the RNA polymerase, it is now free to go from the promoter throughout the gene, the operons, the three genes, the LAC-Z, LAC-Y, and I forget the last one. Let me go back. LAC-Z, LAC-Y, and LAC-A, and transcribe a mRNA molecule. Okay, the TRP operon is an example of a repressible operon. So ask yourself again, is a repressible operon usually on or usually off? It's usually on, therefore it can be repressed. The TRP operon contains 5 structural genes for making amino acids for tryptophan. Again, we have the promoter region, the operon region, and we have, EDCBA genes present. Tryptophan is an essential amino acid. However, when there is plenty of tryptophan around, there is no need to synthesize more. E. coli can just take tryptophan from the environment instead of trying to synthesize it itself. Therefore, the cell uses a repressible operon, not an inducible operon. Remember, that was for inducible genes. This one is a repressible operon in order to shut down production of tryptophan when tryptophan is not needed. So in this situation, we have an inactive repressor protein most of the time. And the RNA polymerase is allowed to read through and create tryptophan, the genes in the metabolic pathway for synthesizing tryptophan. And you get transcription. However, when tryptophan is present, it becomes a co-repressor. That co-repressor will bind to that inactive repressor protein and make it an active repressor protein. And as a consequence, the RNA polymerase cannot move past the operator to transcribe the sequence of structural genes.
Checkpoint 3. In E. coli, the L-arabinose operon consists of 3 structural genes encoding enzymes for breaking down the sugar arabinose. These genes are only transcribed when arabinose is present. Otherwise, the operon is off. The L-arabinose operon is blank.
So now we're going to switch tracks a little bit. We're going to apply what we learned about microbial genetics to viruses and genetic transfer. You may recall that viruses can occur as DNA viruses, where their genome is stored as DNA, or RNA viruses, where their genome is stored as RNA. DNA viruses can exist as single or double-stranded DNA viruses, and they transcribe their genes and replicate their genomes in the host cell nucleus. The viral RNA is translated in the cytoplasm, and all the viral components are assembled to maturity in the nucleus. So how does this occur? Well, the viral DNA enters the nucleus, and early genes are transcribed in the nucleus. Early genes are like the first set of genes that need to be transcribed for the virus copies to be made. The DNA is replicated and RNA is translated. And then finally, the late genes are transcribed and translated. These are genes that have to do with the final touches on producing more viruses. And then these virions are assembled and released. There are several important DNA viruses that we need to be familiar with. These virus families include the Adenoviridae, which cause acute respiratory diseases, including the common cold. We have Poxviridae, which gives rise to diseases associated with skin lesions, including smallpox and cowpox. We have Herpesviridae, which causes diseases associated with sores and rashes, including cold sores, chickenpox, mononucleosis, and roseola. Roseola is a viral illness that most commonly affects young kids between 6 to 2 years of age. It is also known as the sixth disease, but we won't go into that in any greater detail. We also have the Papoviridae, which causes diseases associated with warts and tumors and may cause certain types of cancers. And then Hepatinaviridae, which causes hepatitis B, which is unrelated to other types of hepatitis.
Checkpoint 1. What important family of DNA viruses is known to cause acute respiratory diseases?
Again, not all viruses are harmful, and in fact, some viruses are being used by scientists to deliver certain drugs or gene therapies to humans. The first gene therapy approved for inherited diseases got its first FDA approval for Leber congenital amaurosis, also called LCA, which is a rare inherited eye disease. One of the types of LCA is treatable by restoring a bad mutation in the RPE65 gene. However, the trick is trying to deliver these genes inside of the cells of the eye To do so, scientists used a non-disease causing form of the adeno-associated virus, and this virus can infect the cells but is not disease causing. RNA virus genome structure is similar in DNA virus genome structure in that it can occur as double-stranded or single-stranded RNA. When it occurs as single-stranded, it can either occur as the template strand or the coding strand. If it's the template strand, we call this the antisense strand. If it's the coding strand, if it looks like the coding strand or mRNA, we call this the sense strand. The best way to remember this is that the sense strand makes sense to the ribosome. The sense strand is also called the positive strand, and the antisense strand is therefore called the negative strand. In this image, we can see how those strand naming conventions play out. So again, the top strand is going to be written from 5' to 3', and the bottom strand is written 3' to 5'. Here I've written the start codon first as AUG. If this was DNA, it would be ATG. And the stop codon here is UGA. So you can see that this top strand, which would be a strand that would be recognized by the ribosome, is called the sense strand or the positive strand. And the template strand on the bottom is called the antisense strand or the negative strand. So single-stranded RNA viruses can be further categorized as either being sense strand or antisense strand RNA viruses. Again, that template strand is used as a template for transcribing DNA to mRNA, and the coding strand, which would be complementary and parallel to the mRNA strand, the mRNA strand being that which codes for proteins.
Checkpoint 2. What strand can be used directly for protein synthesis? Meaning it can be sent right to the ribosome and be used as the strand to synthesize the amino acid sequence.
So here are some examples of important RNA virus families to be aware of. The first one is Picornaviridae. This is a single-stranded sense RNA virus, and they can cause the common cold or poliomyelitis. The next one is Togaviridae. This is a sense single-stranded RNA virus, and it can cause viral encephalitis, rubella, and AIDS. Next is the Coronaviridae, and this is a sense-stranded, single-stranded RNA virus, and it can lead to respiratory illnesses. And an example of this virus is coronavirus 2, also colloquially just known as coronavirus. Next is Filoviridae, and this is an antisense single-stranded RNA virus. It can cause hemorrhagic fevers like with Ebola. Then we have Orthomyxoviridae, which is also antisense and causes the flu with influenza. And finally, Rioviridae, which is double-stranded RNA virus and can lead to gastroenteritis. So now that we've discussed the different types of RNA viruses that exist out there, we need to keep track of a few things to understand how virus replication occurs based on the type of RNA virus it is. One thing we need to be aware of is that every new virus is going to need to make RNA and protein, go through the process of transcription and translation, or at least translation. For positive stranded RNA viruses, their positive strand of RNA can be directly used to make proteins. If you need to make more copies of the positive strand, you can only make the opposite copy. So first, they have to make a copy of their positive strand into the negative strand. and then that can be turned, then read to write more positive strands of RNA. The same is true for negative stranded RNA. So negative strand RNA, the antisense strand of RNA, in order to make more copies of that minus strand of RNA, first you have to make the positive strand of RNA. Because remember, these polymerases always read the opposing strand to make a new strand. Right? So if we look at this polymerase here, it's going to be reading the opposite strand in order to make a new strand of DNA. However, we need to also understand that only a sense strand of RNA, essentially, the sense strand is the coding strand of RNA, same thing, can be used to make proteins. So for positive strand, single-stranded RNA viruses, they can use that directly for making proteins, for translating. But for antisense single-stranded RNA viruses, they have to make a positive strand or a sense strand in order to do any translation. This figure kind of depicts just that. Here we have a single-stranded positive or sense RNA virus, and so its genome can directly be translated into proteins. And in order to replicate itself, it's got to make a antisense strand, which acts as the strand that is read by a polymerase in order to write more of its genome, which is the sense strand. And for the negative or the antisense strand or the negative strand, the opposite is true. You have to make a sense strand in order to make proteins, and you need to make a sense strand in order to make antisense genome strands. The trick in understanding the cycle of replicating new viruses is just to keep in mind that you have to have the complementary strand in order to make more copies of the genome. So for a positive strand, you're going to need the antisense strand to make more copies of the genome. For a negative single strand of RNA, you're going to need the positive strand to make more copies of the genome. Double-stranded RNA, it's a little bit different. In order to make a copy of the double-stranded RNA, a copy of both strands needs to be made. So this goes through that process of semi-conservative replication where each strand is copied, and as a result, you can get positive strands of RNA out of this by copying the negative strand of the double-stranded RNA, and those positive strands can be translated into proteins, and these positive strands can be copied into negative sense strands or antisense strands. You take the two of those and you put them together, and what do you have? You have the genome again, which is double-stranded.
Checkpoint 3. A virus infects a host. Its RNA genome must be transcribed into a second strand before any of it can be translated. None of this transcribed RNA exists in the virus before infection. This virus is an example of a blank virus.
Okay, moving right along, let's talk about retroviruses. Retroviruses are very interesting. They have RNA genomes, but they contain a special enzyme called reverse transcriptase. This enzyme is unique in that it can do the opposite of RNA polymerase. It can convert RNA back into DNA. So this viral DNA can then be integrated into the host genome. So if we look at the viral cycle here, we have first the virus penetrates the host cell. Then, after uncoding occurs, reverse transcriptase converts viral RNA to DNA. The viral DNA then integrates into the host cell genome. Remember, the host cell's genome is always going to be double-stranded DNA. So this DNA then gets integrated inside of that sequence, inside of those chromosomes. Next, a provirus is transcribed and translated to make viral proteins. The viral components are assembled by protease, which is another enzyme, and then these newly made virions are released via budding or lysis. The most well-known retrovirus is HIV, human immunodeficiency virus. And let's talk about what viral budding is. It's pretty simple. You have a viral capsid at the top of this image, and you have the host cell plasma membrane, and you have some viral proteins embedded in that plasma membrane. And that viral capsid pushes its way through the membrane and then gets pinched off so that it has an envelope surrounding the virus. Okay, so now let's talk about genetic transfer in prokaryotes. So how exactly do prokaryotes acquire genetic diversity? So first we know how eukaryotes acquire genetic diversity, right? They do so through sexual reproduction. But prokaryotes reproduce asexually, and they acquire genetic diversity from either mutations in copying their genome. So every time you replicate a genome, errors and mistakes get made, and those are called mutations. But prokaryotes have another method called horizontal gene transfer. So what is horizontal gene transfer? This refers to the passage of genes from an organism to, vertical gene transfer refers to the passage of genes from an organism to its offspring. So this would be like parents giving their genetic information to their offspring. And you can imagine that's vertical. If you imagine like a family tree, it passes on down. However, horizontal gene transfer does not require parents or offspring. This refers to passage of genes between organisms of the same generation. And it is thought that horizontal gene transfer contributes significantly to the rise of antibiotic resistance, as mutations that confer resistance are passed between the cells of pathogens. There are three modes of horizontal gene transfer. There is conjugation, transformation, and transduction. Conjugation sounds like what it is. You have a process in which conjugative plasmids cause the production of what is known as the sex pilus, which is this tube which connects two adjacent bacterium to one another through direct contact, creates a direct contact, and genetic material can be transferred through that sex pilus. Gram-negative bacteria have conjugative plasmids that contain the genes that encode a sex pilus. However, gram-positive bacteria do not contain these plasmids. Instead, they produce a sticky surface of molecules that form a mating bridge, which DNA transfer occurs through. Okay, so this figure depicts what this looks like. A conjugative plasmid, known here as the F plasmid, is contained in the donor cell. So the donor cell creates the sex pilus, which makes a direct cell contact with another recipient cell. The F plasmid cell is called the F positive cell, and the cell without this plasmid is called the F minus cell. After this bridge is made, a copy of the plasmid is made. However, only one strand is synthesized, not both strands. And this creates what is called the rolling circle replication, where only a single strand is replicated, produced, and transferred to the new bacterium. Once it reaches the new bacterium, this new bacterium makes the second strand that is complementary to the first strand and circularizes that DNA. So we get, again, a circular, small, double-stranded plasmid. And at this point, both cells become F positive. There are certain types of genes that are transferred by plasmids. If you remember from the beginning of the semester, we discussed that plasmids contain genes that are not absolutely necessary, that they are beneficial for certain environmental conditions. But under normal conditions, normal being in quotation marks here, these cells should be able to survive perfectly well without the plasmids. Some of these genes that are coded by the plasmids include genes for virulence factors. Virulence factor is something that makes a pathogen more pathogenic. So these could be genes that code for verotoxin, which causes dysentery. From the S. dysenteriae, these could be transferred to E. coli. You could have genes that encode for tetanospasmin toxin, which causes tetanus, from C. tetani. You can also have genes that code for antibiotic resistance. So genes for methicillin resistance could be transferred to Staphylococcus aureus and create MRSA, methicillin resistant Staphylococcus aureus. And you can have genes that code for antibiotic resistance to vancomycin resistance, and then you would get VRSA. The next form of horizontal gene transfer is transformation. Transformation is a very basic process. It's the process at which what we call naked DNA, or DNA that's not in a cell but just free floating in the environment, is being passed between cells. So in this figure, we see that here is some naked DNA from a donor cell at one point, and the recipient cell takes up this DNA strand, and then through a process called genetic recombination, it recombines the two strands together, the two pieces of DNA together, to make one piece of DNA. So here we can see that this naked DNA gets absorbed into the cell and then integrated into the genome of the recipient cell. Next, we have transduction. Transduction is very interesting in that it occurs through a mediator. So transduction is the process of transferring DNA from one cell to another through a virus called a bacteriophage or phage. This can also occur in eukaryotes, but here we're focusing more on it happening with bacteria. So a bacteriophage is just a virus that infects bacteria. And so as we know, bacteriophages land on the bacterial cell. They inject their nucleic acids, their chromosome, into that cell. And when the virus makes new viral particles, it needs to package back up new copies of its chromosome. And sometimes it makes a whoopsie and it accidentally packages some of the host bacteria's DNA into its viral particle. And sometimes those viruses that make a whoopsie, they then reinfect a new bacteria, and lo and behold, some of that genetic information can go through genetic recombination. And much like we saw with the previous slide here, where we had transformation after genetic recombination, we get it again here with the host cell. Okay, so we've discussed kind of the three different types of horizontal gene transfer, we have conjugation, transformation, and transduction. And we want to go back and focus on conjugation a little bit. And from what you've learned, you should be able to answer this checkpoint 5. Would you expect the cocci or bacilli in this sample to form a sex pilus for conjugation? This is, I'll give you a little hint, this is a Gram stain. Okay, that's the end of this presentation. Thank you so much for joining me, and I'll see you next time.